afternoon, everyone. It's great to be able to be here and tell you about PTI's work on Alzheimer's disease that we've done over the last 9 months to look at novel protein dysfunctions. Now, I'm sure all of you are familiar with Alzheimer's disease, but I'll give a little bit of background just so we're all on the same page. You probably know that Alzheimer's disease is a relentlessly progressive, fatal neurodegenerative disorder. And perhaps you've even seen an MRI image like this, where if we look at a healthy control on the left and an Alzheimer's disease patient on the right, what is immediately evident is this striking atrophy of the tissue in the Alzheimer's disease patient. And that reflects widespread loss of neurons from throughout the brain in Alzheimer's disease. And unsurprisingly, that widespread neuronal loss is associated with a number of cognitive symptoms. Perhaps the most well-known is memory loss, but we also see things like emotional impairments and cognitive impairments in AD patients as well. So Alzheimer's disease affects at least 30 million people worldwide. That number is expected to at least double by the year 2050. We had no effective treatments for Alzheimer's disease. We had no way to prevent the disease. And so there's an urgent need for new discoveries in Alzheimer's disease to help us tackle this really immense problem that affects so many members of our society. And we think that PTI is especially well positioned to make those new discoveries in the space of Alzheimer's disease, because Alzheimer's and a lot of related neurodegenerative diseases are fundamentally disorders of protein dysfunctions. So pathologically, Alzheimer's disease is defined by two protein features. The first are extracellular aggregates of amyloid beta peptide. They assemble into these hairball-like clumps and accumulate in extracellular space. And we call those amyloid plaques. You can see them in the brains of an Alzheimer's patient. The other feature that defines Alzheimer's disease pathologically are these intracellular aggregates of the microtubule-associated protein tau. So tau is normally bound to microtubules in Alzheimer's disease. It dissociates from those microtubules, forms these intracellular filamentous aggregates. And those occur widely within the neurons of AD patients. You see them in dark brown in the top left there in an actual AD patient brain. So those features are actually how we diagnose AD pathologically. They define AD, and in some ways they've defined a way to research AD as well. We focus extensively on amyloid beta and tau throughout the history since we've known about these proteins and their role in disease. But something I'd like to call your attention to is a theme that's emerged over the last few decades in AD research, which is that protein dysfunctions are actually much more widespread in Alzheimer's disease than simply amyloid beta and tau. So this is a non-exhaustive timeline of other proteins that we now know form pathological aggregates in Alzheimer's disease. And you can see that number has steadily increased over time. But I would also highlight about this list, in addition to the progress it shows that we made, is that most of these proteins were discovered in AD using targeted approaches. And that is, we had a very specific hypothesis about this protein's role in disease, and we studied it individually in the context of Alzheimer's disease. And it's against that backdrop that I want to talk about what we hope to do at PTI to move forward our understanding of Alzheimer's disease. So I'll highlight two goals. The first is to comprehensively characterize protein dysfunctions in Alzheimer's disease. We know that even within a single Alzheimer's disease patient, there are numerous mechanisms that might give rise to protein dysfunctions. And again, a non-exhaustive list is highlighted here. And so to grapple with the complexity that AD poses at the individual level, at the population level, we need to be able to measure many patient samples. And because we can have protein dysfunctions arising from all these different mechanisms, they may touch many different proteins in a different brain. And so we need to also be able to measure proteomes comprehensively in our studies of AD. The second goal that I would highlight for our AD research is to understand cell type selective vulnerability and disease progression. So these protein dysfunctions that I've been discussing, they don't occur in the same place in AD brain, and they occur with remarkable heterogeneity. And I think it's well illustrated this schematic here, which shows the origin and spread of tau pathology here in A, and then amyloid beta pathology here in the Alzheimer's disease brain. And you see that they originate in different places, they spread to different places in different ways. But even if we look at a very well-defined, anatomically precise and well-defined brain region, even within that single region, there is also a spectrum of vulnerability to these protein dysfunctions. There's a spectrum of vulnerability to whether neurons die or not. So in the schematic that I'm showing here, you see the 6 layers of the cerebral cortex arranged on the y-axis here. And then within those layers, you can see specific types of neurons here. And you can see from the color-coding scale here that there's really this spectrum of vulnerability to death in Alzheimer's disease. And so to grapple with these complexities that AD brings with it, we also need to be able to measure proteomic changes within single cells and different cell types. And we also need to be able to make those measurements across multiple brain regions. So what we've done, what we've been doing at PTI is in support of these goals. And I'll talk about what we've done in regards to the first comprehensively characterizing protein dysfunctions in Alzheimer's disease. And for this first part of the talk, I'll talk about some work we've done with pulp JPU samples. Before I get to the work itself, I want to highlight the people who have made it possible. First, the work is an ongoing collaboration with Brett Hyman's lab at Massachusetts General Hospital. A really productive and satisfying collaboration, both in terms of the resources we've been able to share each other, as well as the conversations and the ideas that have come out of collaboration. I want to highlight Corinna Friedrich, our mass spectrometrist. She's done a great job with sample prep for these projects, as also run the data on the mass spec and done a lot of great data analysis. Jason Derks has also helped out with sample preparation and has come up with some really creative analysis that has helped us interpret our data. And that latter thing can also be said for Megan Elcheikhali, our computational scientist, who's also really helped us and analyze this data and come up with ways that we can interpret it that are biologically meaningful. We started with a cohort of 24 samples of AD and non-AD patients. We're profiling a region of the brain known as the dorsolateral prefrontal cortex. It's known to be bald in Alzheimer's disease. And our mass spec approach that we're using is the Plex-CIA approach that we've heard about throughout the day. Now, I'd point out that if we're using the abstract labeling approach here, we've heard a lot of exciting results related to PSM tag today, so you might be asking, how did you use PSM tag? We've been trying to build up our AD research efforts in parallel with the work that's happened on PSM tag. And so this is sort of in some ways our first generation of work that uses existing reagents, but in the future, we're excited to increase the sample size and hopefully the number of proteins that we see by using the tags we've built at PTI. So if we look at our results just at the differential protein abundance level, unsurprisingly, we can see hundreds of proteins that are differentially abundant between AD and non-AD samples, and many of these reflect known changes that occur in Alzheimer's disease. And here our work has benefited from those who have come before us that have done large-scale proteomic profiling in AD brains, particularly the work of Jumin Peng and Nikolai with an alternate interpretation here. But we see things that we know change in Alzheimer's disease, and so we think we have a good data set here. ApoE is the largest known genetic risk factor for Alzheimer's disease. Complement C4a is an inflammation-related protein that's known to increase in Alzheimer's disease. And then conversely, we see decreases in cytoskeletal proteins, another common alteration in AD, and then also changes in signaling pathways. But we wanted to get a more interpretable view of the changes that we're seeing than something like this visualization provides. And in particular, we wanted to understand how proteins are changing within specific compartments of the cells in the AD brain. To illustrate how we did that, I'm just going to provide a simple conceptual example here. We can think about a condition, two conditions, where our cells in condition A have fewer mitochondria than our cells in condition B. And if we do our proteomic profiling approach, on average, perhaps there is no fold change for all of the proteins in the cell. The average fold change here is 0. But if we were to look specifically at mitochondrial proteins and compare condition A to condition B, we would expect to see this leftward shift because there are fewer mitochondria and therefore fewer proteins there. So we, as a sanity check, applied this approach to our proteomic data, and we specifically focused on the cytoskeleton. So I said at the start of the talk that tau dissociates from microtubules in AD, so that leads to tau tangles forming, but it also leads to this collapse of the cytoskeletal structure inside the brains of AD patients. When we look at cytoskeletal proteins in the data that we collected, we see the expected large and significant shifts in cytoskeletal proteins in AD versus control samples there. So since this reflects known AD biology, we use this as basically a scaling factor to look at other compartments within the cell. We expand this analysis out to look throughout the cell, different organelles. And you probably can notice that one of the largest shifts that we see here is with the 20S proteasome core particle. Proteasome is the primary degradation, protein degradation machinery in eukaryotic cells. So we see a big shift in one component of it, but not in another. So to give you a sense of what that looks like, this is what the proteasome looks like, and it consists of two elements. The first is the 19S regulatory particle that binds proteins that are marked for degradation, unfolds them, and feeds them into the proteasome's 20S core particle, which actually degrades the proteins to short peptides. So what we're seeing in our data is an imbalance where the levels of this aspect of the proteasome are decreased. These seem to be unchanged. And we thought that suggested to us that there are imbalances in the stoichiometry of this complex, which may affect the ability of the cells to carry out quality control protein degradation. And so indeed, if we looked at the correlations of subunits of the proteasome within non-ED cases at the level of both the 20S core particle as well as the 19S regulatory particle, we see the expected very strong correlations among subunits. This sort of correlations we'd see many times across many different systems. On the other hand, if we look at our AD samples, we see significant reductions in the correlation of proteins at the 20S core particle, as well as an even more significant reduction within the 19S regulatory particle. So what we're seeing here is a reduced stoichiometry of proteasome subunits, and that may reflect a reduced capacity of cells and AD patients to carry out essential protein degradation activities. And to see how that's affecting the proteins that we see that are differential abundance in our samples, we specifically asked the question, are there proteins that are rising to increased abundance because the proteasome capacity of the cell is decreased? And to address that question, it's important to understand that proteins contain within them these regulatory sequences called degrons. Degrons can be bound by enzymes of the ubiquitin system. And when that happens, the protein becomes ubiquitinated. And then ubiquitinated proteins can be marked for degradation and then bound and degraded by the proteasome. And so we took our list of proteins that we know are differentially bound, including our samples, and we asked, are they more likely to contain sequences that allow proteins to be targeted for degradation? And indeed, although the proteins that are differentially abundant have similar links, I'm not showing that data here, you can see that proteins that are increased in abundance are significantly more likely to contain a degradation signal than those that are of decreased abundance. We then asked, are there specific types of degradation signals that are in our proteins of increased abundance? On the x-axis here, I'm showing the ratio of proteins that are of increased versus decreased abundance. And then on the y-axis, I'm showing specific types of enzymes that recognize specific degradation signals. And you can see that there's a large and significant increase in this BAG6 complex. This result was intriguing to us because BAG6 is actually a quality control protein chaperone. It recognizes proteins that are mislocalized, proteins that rise to abundance perhaps because they can't be properly targeted for degradation. So we took our list of proteins that are recognized through this quality control mechanism, this BAG6 complex, and we asked what are the proteins that are of increased abundance there. And what stood out to us is this protein TMAM94. It's an ER-resident protein, but it also contains two of these BAG6 recognition motifs that allow it to be targeted for proteasomal degradation. And so because it contains these recognition motifs and because it's recognized by something that specifically targets mislocalized proteins, we think that this may be rising increased abundance through this decreased proteasome capacity in the cell. And indeed, we think that these recognition motifs that you see here are authentic BAG6 motifs. In a previous high-throughput assay that measures degron strength, you can see that as compared to the rest of the BAG6 N-terminus, they're significantly stronger in their ability to catalyze degradation, suggesting that they're true, authentic BAG6 motifs. So we asked, in addition to the proteasome, this proteasomal defect that we measured in 90 samples, might the targeting of TMAM94 be affected by the stoichiometry of this complex as well? And indeed, if we look at the correlations of the members of this bag-6 complex, you can see that they, like the proteasome, are significantly reduced in our AD samples relative to non-AD samples. So we think that this is a very specific protein dysfunction that's arising through quality control mechanisms that are likely impaired in Alzheimer's disease samples. And this is an area that we're really excited to follow up with more mechanistic work through our collaboration with Brad. So that's the work that we've done to characterize protein dysfunctions in AD. I want to talk about the second goal of our research, which is to understand cell type selective vulnerability and disease progression. And as I said at the start of the talk, for that we really have turned to single-cell proteomics. And we want to do single-cell proteomics on cells that we derive from mutant-grade fission. And before I get into our work to that end, I want to specifically highlight what Peyton Stewart has done to really move these projects forward. He's worked immensely hard and come up with a number of creative solutions that have really driven the research forward that have supported this effort. And so isolating single cells for human brain tissue poses significant challenges, as you can imagine. Although conceptually what we want to do is very simple. We want to go from this to this. In practice, we'll perhaps undershoot it, as you can see here, or we can also overshoot it. And perhaps more significantly, the approaches that we have commonly used to dissociate cells tissue to cells are manual in nature. So there's likely issues of how much can we scale this, as well as how reproducible is it from person to person. You can also, an alternative approach is to use these protease enzymes. Those perhaps work well in the context of single-cell RNA-seq, but working at the level of proteins where these enzymes may have non-specific activities, it also poses challenges. So what Peyton has come up with, working together with folks from Nikolai's lab at NU, is an approach that is non-enzymatic and that's fully automated and standardized. We use this device that allows us to slice and grind tissue, that you can see here. And the result of this is that we get these morphologically identifiable neurons that have these nice processes that allow us to say, indeed, this is a really nice neuron that we've been able to isolate from this human brain tissue. Of course, it's one thing to see these nice-looking neurons underneath the microscope. It's another to be able to collect them. And so our approach for this is using the NPOP approach that was developed at Nikolai's lab at MU that we've heard about today. And we use an instrument called the Salmon 1, developed by Scineon, to collect cells. And so one of the things that's been really stunning with this project is to be able to not just see really nice neurons at the level of the microscope, but also to see them coming through the instrument as we're collecting them. So you can see we still have that nice neuronal morphology. We have projections that we can see that survive the process of being flowed through here. And we also can tell that these are neurons by staining for the nuclear marker, DAPI. And so with this, we can combine this fully standardized automated approach that we've come up with this NPOP-based approach to prep thousands of cells, thousands of neurons that we've collected here. And I would emphasize here, too, that the approach that we've taken is to focus on neurons. And we acknowledge that approach has trade-offs. There are, of course, non-neuronal cell types in the brain as well, and those have interesting and probably important roles in diseases like Alzheimer's. But we also think that neurons are of clear biological interest in the context of AD, and that our ability to hone in on them makes this a really enticing area for this first single-cell proteomics project of AD and PTI. And so to this point, we've collected around 5,000 cells from across the stages of Alzheimer's disease. We get really nice distributions of cell size across the stages here, suggesting that we indeed are capturing neurons from throughout the stages of Alzheimer's disease. And more excitingly, we're getting what I think is really nice proteomic data from these cells. We get around 2,300 proteins per cell, and excitingly, we get the big players, proteins that are of known relevance in AD, so tau, alpha-synuclein, this amyloid beta precursor protein, as well as TDP-43. So we're getting lots of proteins. We're getting proteins that are of known interest in the context of AD as well. We've begun to start looking at data completeness, and we're also seeing encouraging results on that side of things. And then more recently, we've begun to look at how intact the cells that we are collecting are. And to do that, we're looking at what we're essentially looking for is protein leakage. So to what extent do the proteins from various compartments in the cells appear to be intact relative to a bulk sample that we might profile? And so you can see some analysis that Jason carried out for these single cells that we've collected. You can see on the x-axis is sort of the log2 fold change for the single cell data that we have versus the bulk. And you can see that overall, it depends on which protein you specifically look at, but we think that there's relatively little depletion from these samples. And I would say that these analyses are relatively preliminary. Data is literally rolling off the mass spec as I'm giving this talk, so we're still in the early stages here. But I think you can see one thing that is maybe not obvious from the data is that I think these distributions will tighten up as we look at more proteins and really build out these analyses. But preliminarily, we think we have relatively intact cells from this process that can be a really rich resource for AD research. So to wrap up here, I've highlighted a couple of goals for our AD research at PTI, and these are very much goals that we're in the midst of completing right now. We think we're getting the data. We still have more data to collect that can really build out and support the analyses that we want to do. But at the same time, I think the work that we've done so far has also shown that we can indeed find new protein dysfunctions that are occurring in Alzheimer's disease by generating high throughput proteomic measurements that are comprehensive. So I'll specifically highlight the changes in the 20S and 19S regulatory particle subunits that we've seen in our bulk samples, as well as this change in this TMEM94 protein that is potentially mislocalized in AD. And then I think we also have a very clear path forward to understand cell type selective vulnerability using single-cell proteomics that combines our approach for isolating morphologically identifiable neurons and the single-cell proteomic methods that are being developed at PTI. And of course, these goals are not independent of each other. We think that they support each other, and achieving both of these goals will be an important step forward to having a better understanding of Alzheimer's disease. Of course, with an interdisciplinary project like this, there's lots of people to thank. But first and foremost, we want to thank the patients and their families. They're the ones that make this research possible through their donations of samples. So we're immensely grateful for the opportunity to work with those. And then there's a few specific people to highlight here. At the PTI level, the mass spec team has helped us really get great data. Thibaut and Lena have helped us learn to use the Salem-1. And Alex, I would highlight Alex Petelski, who kind of kickstarted all of our AD research efforts at PTI. She's since moved on to an independent position. All the team at MGH always has great suggestions. They've been a rich resource for us. Andrew and Sina and you have really helped us with the sample prep side of things. I appreciate of that. And also on the sample prep side, Josh Gantlin has helped us a lot as well. So we're appreciative of all that help, and I'm happy to take any questions.
All right, thanks, Mahlon. Let's open it up for some questions.
Thank you. That's really interesting work, especially with the Alzheimer's degradation pathway dysfunction that you discovered. And specifically, do you think you've potentially identified a mechanism that can be targeted with maybe some sort of drug with it?
Yeah, I think there's a lot of interest not just in the context of Alzheimer's, but generally in how one can amplify these quality control pathways, right? So like alternative fasting was kind of the fad thing. And one of the hypothesized mechanisms there is that you're forcing your cells to do autophagy, which is sort of another route for protein degradation. So I think generally there is a lot of interest in increasing the activity of these clearance pathways, because we know that as the brain ages, there are multiple things that sort of accumulate more and more damage, and that leads to sort of an increasing loss of protein homeostasis that occurs over time. So I think that is sort of a fundamental mechanism of aging and also age-related diseases that I'm sure lots of folks are working on targeted therapeutically.
It's sort of following up on the blackout of protein homeostasis in the 80s. So if you look at other complexes not involved in degradation, do you also see them dysregulating? If you look at metabolic complexes or chaperone systems, is it just generally like proteostasis is broken now all these complexes have dysregulated?
Yeah, we haven't done a deep systematic analysis of protein complex homeostasis within the disease, but my expectation would be that that is the case, right? So these there's multiple pathways within the cell that are specifically dedicated to eliminating excess subunits of complexes that are produced, and so with the proteasome being compromised in these cells, with ubiquitin ligases also the stoichiometry being not there, we expect that those sort of excess subunit accumulation things are happening. And all of these things are sort of accelerating the decline of protein homeostasis is happening in disease, right? So, so our expectation is certainly that those are altered as well.
Thank you. I'm wondering if, in the case of Alzheimer's, if you're going to look at other cell types in addition to neurons. Yeah, certainly other brain cell types have been implicated. Yeah.
Yeah, absolutely. So one of the things I would say up front is that the approach that we've developed, we think, allows us to target any cell type that we want, right? So we can add fluorescent markers in. It's like you have a big marker for your astrocytes, your oligodendrocytes, things like that. We can go after those populations as well. The choice of neurons is somewhat based on the current throughput of the technologies that we have right now. So we think at current throughputs, a sort of because one can run up the tissue here, and I'm showing that we can get neurons from this, but we think that there's probably also intact astrocytes, oligodendrocytes, other cell types in there as well. The question is, can you sample enough of that to get enough of a certain cell type to be able to analyze it? And so we took data from the Allen Cell Brain Atlas, and we just looked at the abundance of cell types that they see, and we said, okay, if we sample at our current throughputs, what do we have the best chance of success with? And there the answer was very clearly neurons. But certainly going after cell types like astrocytes in a very targeted manner, we think is also something that can be of great value for the AD community.
My question is related. With the selective cell vulnerability, just looking at neurons, is the idea then that you'll go to some type of neuronal subtype, or what happens after you identify those pathologies that aggregate with the neurons?
You're asking, do we want to stratify the analysis further to specific neuronal cell types?
Yes.
Yeah, absolutely. And we think that the data can be our best guide in doing that. So we certainly can get out neuronal subtypes, but actually what we'd like to define is sort of where in the disease process are these neurons, right? So what would be really exciting from our perspective is to be able to kind of stage the neurons on sort of like a pseudotime axis, where we say, okay, this population of neurons is relatively early in disease. These are sort of at the midpoint, and these are about to die here. And then seeing how the protein was changing across that axis, we think can be really informative, because we certainly have a good handle on what a dying cell looks like. I'm not sure that we have as great a handle on what a cell that's kind of early in that death process looks like, particularly within an actual human brain, as opposed to cell additions.
Any more questions from the audience? We do have one online. How applicable might this single-cell data collection approach be to study other neurodegenerative diseases?
Very, very applicable, right? So, and our ability to selectively isolate neurons, I think, is really exciting in the context of other diseases like ALS. So in ALS, you have a very well-defined population, spinal motor neurons that are dying as part of that, and so you could imagine taking spinal cord tissue and selectively isolating motor neurons that way. We think it can be relevant for that. I would also add that these diseases, Alzheimer's, Parkinson's, and ALS, they actually share a lot of pathology. So like alpha-synuclein pathology is hallmark for Parkinson's, but it also occurs in a lot of AD patients as well. And so looking across the spectrum of these diseases, I think, can also be useful for understanding the proteomic drivers that sort of generally affect proteins that we know aggregate, and the ones that have very specific effects on something like tau versus alpha-synuclein. So we think the method is broadly applicable to understanding what's happening in neurons that are dying in these diseases.
Thank you. We didn't have one more, but I think you already answered it. It was how scalable is the single cell method to include other cell types. So we addressed that already. So in that case, let's thank Melloni again.